Hello, everyone. Good morning, and welcome to Hunter and I's presentation on hemoglobinopathies other than HBSS and HBC inherited. So what are hemoglobinopathies? Hemoglobinopathies are structural and qualitative defects in the synthesis and configuration of hemoglobin. So there are alpha-like and beta-like genes. Alpha-like are on chromosome 16, and beta-like are on chromosome 11. And alpha-like consist of alpha and zeta, and beta-like consist of beta, delta, gamma, and epsilon. So as we're developing in utero, the expression of the alpha hemoglobin is first for GAR1 and GAR2, and then hemoglobin Portland, and those are slowly replaced by the the gamma chains, so to create fetal hemoglobin. And then after that, gamma chains are decreased and replaced by beta chains to develop adult hemoglobin, which consists of alpha 2 and beta 2 chains, which is the normal hemoglobin, which is about 95% of our hemoglobin is made of HbA. And then the delta genes are also activated during childbirth for the secondary adult hemoglobin, which consists of alpha 2 and delta 2. And normal adults have 3.5% of HbA2 and about less than of that of HbF from 1% to 2%. So hemoglobinopathies, they vary in their zygosity, so genetic mutation, and pathophysiology, genetic manifestation. The genetic mutation that causes the most severity and symptoms is usually the inheritance of homozygous mutated alleles from each parent, so it's intrinsic. This causes some kind of anemia, splenomegaly, fatigue, bone marrow failure, vaso-occlusion crisis, and organ failure. And the first hemoglobinopathy we'll talk about is hemoglobin E. So it is usually found in Southeast Asia and is actually growing in prevalence in the United States due to immigration. It is found a lot in Thailand, where it's thought that it can help defend people who have it from malaria. So it is a qualitative and quantitative with a beta thalassemia phenotype. It's usually due to the amino acid defect and the decreased production of the globin chain. So the HB is a beta chain variant in which lysine is substituted for glutamic acid in position 26, which results in a net charge of 2+2, but because of the position on the hemoglobin, it does not polymerize, so there is no sickling of the cells. Homozygous HBE manifests as a mild anemia with microcytes and target cells. It has no observable icterus, which is jaundice, no hemolysis really, and there is usually mild splenomegaly though. It is in 90% people with homozygous patients with 90% hemoglobin E will have a lower MCV, so they're microcytic. In heterozygous patients, the MCV is of 63 fluid femtoliters, so they're also going to have some microcytic cells and slight erythrocytosis and target cells. HBE is confirmed usually with electrophoresis and high performance liquid chromatography. They're microcytic, like I said, hypochromic RBCs, so they have a large central pallor. They're observed codocytes, and in homozygous hemoglobin E, and then erythrocytosis with which is lots of RBCs or red blood cells with target cells and heterozygous hemoglobin E. There isn't necessarily any treatment for hemoglobin E just because it's not as intense as the other hemoglobinopathies, unless it's usually in conjunction with a beta thalassemia. And then it becomes much more severe, resembling beta thalassemia major, which requires frequent blood transfusions. So yeah, and genetic counseling, of course, for whenever you are trying to have children or just for yourself to make sure that you're taking care of your body. So this is a peripheral blood smear of the hemoglobin E, and you can see in here there's honestly a variance in size of the red blood cells. There are target cells present, There are a few schistocytes. That platelet in the middle is very large. There are some stomatocytes that I can see, and you can see how some of the cells have a very large central pallor. They're not very they don't have a strong stain or carry the stain very well. So that is hemoglobin E.
All right, so the next hemoglobin we're going to be talking about is hemoglobin O Arab. So hemoglobin O Arab is a beta chain variant caused by the substitution of lysine for glutamic acid at amino acid position 121. The disorder is primarily found in Kenya, Israel, Egypt, and Bulgaria, and about 4% of African Americans. People with this hemoglobin variant generally don't have any clinical symptoms except for mild splenomegaly in the homozygotes. When HBO Arab is inherited with HBS, severe clinical symptoms similar to those in HBSS can result. Homozygous individuals usually display mild hemolytic anemia with many target cells on the blood film. And they'll have a negative result on the hemoglobin solubility test, as well as, the way to confirm this variant is usually with electrophoresis or HPLC. Hemoglobin O Arab migrates with HbA2, HbC, and HbE. So what that basically means, we also have to use a citrate agar at an acid pH to help differentiate it from HbC. Since there are no clinical symptoms usually displayed, there's no treatment generally necessary for people with this hemoglobin variant. And next one. So you'll see on this slide some of the picture. This picture has some poikilocytosis. So we have some trypanocytes, we have sickle cells, a little bit of caudocytes, and this is just what a blood smear of HBO Arab can also look like. And there are some actually slight sickling of the cells when it is heterozygous for HBS O Arab. All right, the next group is hemoglobin D and hemoglobin G. So hemoglobin D is a group of at least 16 beta chain variants, whereas hemoglobin G is 6 alpha chain variants. They tend to migrate in an alkaline pH at the same electrophoretic position as HBS. This is because the alpha and beta subunits have one fewer negative charge at an alkaline pH than HbA and HbS. They don't sickle when exposed to a reduced oxygen tension. They're generally also named for where they've been discovered. For example, HBD Punjab, HBD Los Angeles, and HBG Philadelphia. Those are some of the other common hemoglobin types. It's generally found in African Americans, people from Ghana, in HBG. HBD, however, primarily affects those from the Middle East. HBD and HBG don't sickle. They produce a negative hemoglobin solubility test. On alkaline electrophoresis, HBD and HBG have the same motility as HBS. HBD and HBG can be separated from HBS on a citrate agar at a pH of 6. These variants should also be suspected when a hemoglobin band is encountered that migrates in the S position on alkaline electrophoresis. It also has a negative hemoglobin solubility test. In the homozygous state, there's a greater than 95% HBD with normal amounts of HbA subunit 2 and HbF superscript 33. HBDD can also be confused with the compound heterozygous state for HBD and beta thalassemia. The two disorders you can differentiate between the two based on the MCV. So HBD beta thalassemia produces a low MCV and an elevated HbA2, whereas both values are usually normal in HBDD disease. Treatment. There's usually no treatment unless it's combined with HBS, and then you're going to want to have transfusion and genetic counseling if necessary. The morphology tends just to be target cells when homozygous and when heterozygous trypanocytes or sickle cells can occur. So here on the right, we have a blood smear of what HBG and D homozygous would look like. So you'll note the podocytes, and over here on the left is HBSD. So we have some sickling.
So there are some global impacts of hemoglobinopathies on the world. So in underdeveloped countries, when they decrease the malnutrition and spread of infectious diseases, in some areas they will have more consumers of the medical systems that they put in place, putting an induced burden on blood banks and people donating blood. So a transfusion therapy can cost as much as the whole medical system in itself. So in the future, whenever we are considering blood transfusion therapies in places in underdeveloped countries, they should definitely have a jump on gathering the blood that they need for these people with hemoglobinopathies and thalassemias, because it is fairly prevalent within the world's population. Thank you for joining us.